Hello students, today we are going to discuss about a new chapter that is cell cycle and cell division, right? As you know, the growth and reproduction are characteristics of cells, indeed of all living organisms, right? All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, and ultimately giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells, right? So that means growth আৰু reproduction প্ৰত্যেক সেলৰ এটা characteristics হয়. Indeed প্ৰত্যেক living organismsৰ growth and reproduction এটা characteristics হয়. So growth and development হ'বৰ কাৰণে ultimately আমাৰ কি হ'ব লাগিব, cell division হ'ব লাগিব, যাৰ থ্ৰুৱেদি তোমাৰ এটা organism grow কৰিব পাৰিব, right? Now if you see the definition of cell cycle, then the sequence of events by which a cell grows its size, right, synthesizes all its biomolecules, replicates its genome or genetic material, duplicates the number of cell organelles, right, and eventually divides into two daughter cells is termed as cell cycle. Tar mane eta cell cycle কি হয়, cell cycle এটা sequence of events হয়, right? এটা কিছুমান ইভেন্টৰ এটা sequence হয় য'ত যোনটো cycle য'ত সেলটোৱে তাৰ নিজৰ সাইজটো ইনক্ৰিজ কৰিব, দেন তাৰ যিবিলাক কনষ্টিটুৱেন্টছ থাকিব, লাইক বায়োমলিকুলছ যিবিলাক থাকিব সেলৰ, সেইবিলাক সিনথেসাইজ কৰিব, তাৰ জিনোম ৰেপ্লিকেট কৰিব, yes, DNA, that means genetic material duplication হ'ব, right? তাহাঁতৰ সেল অৰ্গেনিলি ডেভেলপ ডুপ্লিকেশ্যন হ'ব, right? And eventually দুটা ডটাৰ সেলত ডিভাইড হৈ যাব, আৰু সেই গোটেই প্ৰচেছটো কোৱা হয় সেল সাইকেল, right? Now if you see this flowchart here, so the cell cycle can be divided into two phases, right? Interphase and mitosis, right? And interphase is divided again into Z1, Z0 or the quiescent phase, S phase or Z2 phase, right? Z1 phase is the first growth phase or the first gap phase, and the Z0 phase is the quiescent stage, right? And S phase means the synthetic phase, and Z2 is the second gap phase or second growth phase, right? And mitosis phase we can divide it again into two substages, right? So one is your karyokinesis and another is cytokinesis, right? Karyokinesis and cytokinesis. Now karyokinesis means the nuclear division, right? So karyo means the nucleus, right? And division of nucleus is karyokinesis, and this karyokinesis is again divided into four substages, right? So prophase, metaphase, anaphase and telophase, right? Now after the division of the nucleus, cytoplasm division will take place, that is called cytokinesis, and ultimately cell divides into two, right? Now why do cells divide? So cells divide hoy, cell kyo divide hoy? Because there are certain reasons, right? So number one is the decrease in nucleocytoplasmic ratio, right? The change in the nucleocytoplasmic ratio can induce a cell to divide. Next is your replication of DNA, leading to doubling of the DNA content. যেতিয়া সেলৰ ভিতৰত DNA content double হৈ যাব, DNA replication হ'ব, তেতিয়া কি হ'ব, সেলটো ডিভাইড হ'বৰ কাৰণে সেলটো ইনডিউস কৰিব, right? কেতিয়াবা কিছুমান factors থাকে mitogens বুলি কোৱা হয়, right? So which are also known as mitosis promoting factor. So he mitosis promoting factor বিলাক যেতিয়া synthesis হ'ব, right? The synthesis of these mitogens again, so it will induce the cell to divide, and it may be due to the effects of mutations, right? Mutations means change in the sequence of DNA, right? Genetic change as seen in many cancer cells. So these are some reasons due to which a cell divides, right? Now if you see the interphase, now if you see this cell cycle, so here the G1, S and G2 phase, these three together constitutes the interphase, right? Interphase is a non-dividing phase of cell cycle which was earlier known as the resting phase of cell, where it prepares for division. That means eta cellor pora jete duta cell hobo tar division hobo, so tar karone gote preparatory stage to hogol interphase, right? It is a biosynthetic phase where a cell performs intense metabolic activities that include your synthesis of DNA, RNA, protein, enzymes, as well as energy-rich molecules like ATP. Okay, and this interphase, that means these three phases constitute almost 95% of the cell cycle. 95% of the cell cycle, right? Now G1 phase, this is the first phase of interphase, right? So if you see this diagram, so this one is your G1 phase, right? And this is the first phase of interphase, right? Now G1 phase is the longest phase of interphase occurring between mitotic phase and initiation of DNA replication, right? It is the longest phase of the interphase, right? The G1 phase, it is called as first growth phase since it involves the synthesis of RNA, proteins and membranes, which leads to the growth of nucleus and cytoplasm of each daughter cell towards their mature size, right? During G1 phase, the cell is metabolically active and continuously grows, but it does not replicate its DNA, right? So in G1 phase, it does not replicate its DNA. DNA synthesis no. Tar bahire baki amino acids, lipids, proteins, right? RNAs, gute bostu khini yat synthesize hobo. So this is a time of resumption of the normal cell metabolism which has slowed down during the previous cell division, right? Thus, the G1 involves transcription of three types of RNAs, right? rRNA, tRNA and mRNA, right? And the rRNA synthesis is indicated by the appearance of nucleolus in the interphase nucleus, right? And proteins synthesized during G1 phase are certain regulatory proteins, right, which controls various events of mitosis, and certain kind of enzymes, right, like DNA polymerase, which are necessary for DNA synthesis in the next stage. That means in this S phase, right? So this one is your S phase. Okay. Then this G1 phase, it occupies almost 30 to 50% of the total time of the cell cycle, right? And in some cells which are rapidly dividing cells, right, like blastomeres of the early embryo of frogs and mammals, right? So they entirely lacks the G1 phase. So that's why the G1 phase is most variable phase of the cell cycle, right? Next is your G0 phase or G0 phase or quiescent phase. So once a cell completes the G1 phase, the cell is committed for cell division. Now it is committed for cell division, right? If a cell skips the cell cycle from G1 to carry out various functions and differentiates into specific cell types, the cell is said to be entering into an inactive stage called Z0 or Z0 or quiescent phase, right? So the cells entering this phase do not divide but remain metabolically active, right? So the exit of cell cycle into Z0 or Z0 phase is temporary, as seen in suspended parenchyma cells, which regain capability to divide into mature stages to form meristematic cells in plants, right? Or permanent, as in case of neuron, right? Heart cells in adult human, and sclerenchyma cells, xylem vessels forming the cell in plant, right? Now if you see, the next phase is the S phase or the synthetic phase. The most important event of S phase is replication of DNA or genetic material. This is very, very important because S phase is marked by the synthesis of DNA or the genetic material, right? It is important to note that the DNA content inside a cell doubles from 2C to 4C, right? But the number of chromosome remains same. This concept of C value we'll discuss it later, right? But the number of chromosomes remains same. Here the content of DNA increase, but the chromosome number remains same. As such, chromatins appear as diodes at the end of the S phase without increasing the chromosome number. This point is very, very important, right? Now in this phase, centriole duplicates in cytoplasm of animals and become ready to move to the opposite pole. Here in this phase, the synthesis of histone protein is an important event, right? So histone protein is required for the DNA to wrap around it, right? So that's why histone forms the core, right? So around which the DNA it wraps, right? And the assembly of kinetochore subunits with respective chromosome is seen in this phase. That is your S phase, right? It is marked by, important thing is that it is marked by the period during which the DNA synthesis takes place, right? In animal cells, during S phase, DNA replication begins in the nucleus and the centriole duplicates in cytoplasm, right? And plant cells, they do not have centriole, right? This S phase, it occupies 35 to 45% of the total cell cycle, right? Next is your Z2 or second gap phase or the second growth phase. It is the period between the synthetic phase and the mitosis, right? Period between mitosis and the synthetic phase. And as such, this phase is also called post-synthetic or pre-mitotic phase. This phase is initiated when DNA replication stops. Yes, after S phase, DNA replication stops and G2 phase starts. But the synthesis of proteins and RNA continues here, right? And most of the cells organelles, yes, most of the cell organelles, they divide or duplicate and almost double their number in this phase, because now in the next step they will enter the M phase, right? The divisional phase, mitotic phase, right? So the remaining macromolecules that are required for cellular division are also formed. One important protein, that is your tubulin protein. This tubulin protein is synthesized here, and which are required for the formation of spindle fibers, in the M phase, right? So it is required for formation of spindle fibers in the M phase, and the cell is fully grown in size at the end of the Z2, and now it is ready to enter into the M phase, right? Next is your M phase, right? Sorry. Now, mitotic phase is the actual phase of division where a cell physically divides into daughter cells. This phase is completed in two phases, right? This phase is completed in 2 phases. This karyokinesis, that means division of the nucleus, and the cytokinesis, that is division of the cytoplasm, right? Now karyokinesis is again divided into 4 substages, right? The nuclear division is again subdivided into 4 phases: prophase, metaphase, anaphase, and telophase, right? And M phase is the shortest period of cell cycle, which is completed in about 1 hour of the entire duration of a 24-hour cell cycle, right? 24-hour cell cycle are 23 hours interphase jai or 1 hour M phase jai, right? So it is important to note that M phase is usually referred to as mitosis phase and not meiosis. This is because the haploid daughter cells of meiosis do not divide further in order to carry out another round of division, right? So this is your all about M phase, right? Now you see the concept of C value. So what is C value? C value is nothing but it is the content of DNA in one set of chromosome, right? In one chromosome, the amount of DNA present, that is called as C value. Yes, that is denoted by C, right? Normally a diploid cell, that means this is the cell, so in which the single copy of DNA has twice. That means one chromosome has 2 copy, right? That's why it is twice n, and the C value is 2C. That means for one chromosome the DNA content is C, so for for 2 chromosomes it is 2C, and after G1 the cell becomes larger, right? The cell will grow in size, but the C value will remain same, right? That is 2C. After S phase, what happens is that it is the synthesis phase, right? So in the synthesis phase, when DNA is synthesized, DNA per cell doubles. That's why the C value becomes 4C. But note that the chromosome number remains same, that is twice n. That means after S phase, the chromosome number remains twice n and the DNA content becomes 4C, right? So it is in the anaphase when the chromosome number is 4n. That means দুটা সেল যেতিয়া ডিভাইড হ'ব from a single cell, তেতিয়া কি হ'ব দুইটা সেলত ইকুৱেল নাম্বাৰ অফ ক্ৰোমোজোমছ যাব লাগিব। সো তেতিয়া কি হ'ব আলটিমেটলি ক্ৰোমোজোম ডিভাইড হ'ব, ৰাইট? আৰু তেতিয়া ক্ৰোমোজোমৰ কন্টেন্টটো হ'ব, ক্ৰোমোজোম নাম্বাৰটো হ'ব 4n আৰু DNA কন্টেন্টটো অলৰেডি ডাবল হৈ আছে। ইয়েস, সো হেতু কাৰণে DNA কন্টেন্ট 4C। দ্যাট মিন্স এট দ্য এন্ড অফ দ্য M phase, DNA content will be 4C and the chromosome number is also 4n, right? So this is the concept of C value. Now if we summarize, then you see that this is the G1 phase here, this is the G1 phase, and here in this phase cell grows and carries out its normal metabolism, right? And in S phase it is the synthesis phase where DNA replication takes place, chromosome duplication takes place, right? And the histone proteins are synthesized here in this S phase, right? Histone proteins are synthesized. And in Z2 the cell grows and prepares for mitosis. That means in Z2 the cell, RNA continues to synthesize, right? Protein continues to synthesize, but here in G2 a specific protein, that is tubulin protein, is synthesized, right? Which is the constituent of the spindle fiber. Yes, and that is why this tubulin protein is synthesized here, so that they can provide the tubulin protein for the production of the mitotic spindle or the spindle fibers, right? And in the M phase, right, so here this one is the M phase, so it is again divided into prophase, metaphase, anaphase and telophase, right? So the telophase marks the end of the M phase, and ultimately the cell divides into two. That means the nucleus divides into two, and after cytokinesis the cell completely divides into two from a single cell, right? So this is the overall picture that is summing up. And now if you see the regulation of cell cycle, that means how the cell cycle is regulated, how it stops, right? How it allows the cell to pass through different stages, then you will see certain set points. And these set points are nothing but these are control points in the cell cycle, right? Where stop and go ahead signals can regulate the cell cycle. There are three important set points: G1, G2 and M phase set points, right? So in these set points there is a special kind of proteins, right, which is known as cyclin proteins. These are required, and there is an enzyme, right, group of enzymes known as cyclin dependent kinase enzyme, right? So these cyclin dependent kinase enzymes are normally inactive, right? When cyclin combines with this cyclin dependent kinase enzymes, then only the kinase enzyme becomes activated, right? And they ultimately checks the whether the cell is synthesizing its constituents, right, in a whether the cell it synthesizes it's properly, the constituents synthesizes properly or not, right? And in the G1 phase there is a different kind of cyclin that is synthesized known as G1 cyclins, and in G2 phase there is another kind of cyclin that is synthesized which is known as M cyclin. So now in the G1 sec points, the cyclin protein will bind with the cyclin dependent kinase enzyme, right? So this cyclin protein, it will bind with the cyclin dependent kinase enzymes, and it will activate this cyclin dependent kinase enzyme, right? And now it will check that whether the protein that are synthesized in the G1 phase is proper or not, right? RNA, ATP molecules, enzymes, right? All these are synthesized in G1 phase are proper or not. If jodi gote bostu khini properly hoy tene hole ki hobo, cell tok allow koribo to go to the S phase, right? Next similarly G2 set point thakibo. So tat ki hobo, cyclin dependent jitu kinase enzyme thakibo, tak again cyclin activate koribo. Jetia e synthesis phase or humai jabo G1 check point or pisot, tetia এই cyclin যেটো থাকে আমাৰ ই কি হৈ যাব, ডিগ্ৰেড হৈ যাব, আৰু ডিগ্ৰেড হৈ যোৱাৰ পিছত ultimately this CDK cyclin dependent kinase enzymes will also become inactive. Again যেতিয়া G2 phase ই যাব তেতিয়া কি হ'ব, আকৌ cyclin synthesize হ'ব, right? এই cyclin গৈ পেলাই আকৌ ইয়াৰ লগত বাইন্ড কৰিব CDK লগত, ultimately activating this cyclin dependent kinase enzymes, and this is known as maturation promoting factor, right? MPF, right? This complex of M cyclin and CDK is also known as maturation promoting factor, right? Now activated CDK cause a transition of the cell from G2 phase to the M phase, right? This is followed by the degradation of the M cyclin at the end of the M phase, right? তাৰপিছত যেটো M phase check point থাকিব, সো তাতে সেলটো সোয়া হ'ব যে সেলটো properly তাৰ spindle fibers বিলাক synthesize হৈছে না, আৰু properly spindle fibers বিলাকে kinetochore লগত attached হৈছে না, right? In the M phase check points, it checks, the cell checks whether the spindle fibers are properly attached to the kinetochore or not, right? So if we sum up, then we'll see that G1 check point, it checks for the damaged DNA, right? It checks for enzymes, proteins, ATPs are properly synthesized or not, right? And this is done by the activation of the cyclin dependent kinase enzymes with the help of G1 cyclin, right? And this caused the transmission of the cell from G1 to S phase, right? So you see here the summary of the events, right, sequence, and the in is in this next sequence, that is in the Z2 phase, the sequence that is present in Z2 phase. At the end of the Z2 phase, it checks for the proper DNA replication, whether the DNA replication in S phase is proper or not. It checks for proper synthesis of proteins, enzymes and ATP, right? Checks for tubulin protein synthesis, right? And M cyclins bind to inactive cyclin dependent kinase here also, right, which is known as maturation promoting factor, and this causes the transmission of the cell from Z2 to the M phase, right? And then finally degradation of M cyclin occurs at the M phase, right? And in M phase sequence, the cell checks whether the spindle fibers are properly attached to the kinetochore or not, right? So this is all about for today. Thank you.